Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Syamim bin Muhammad SHAMSURI and my metric number is 51910. I will introduce about abstract. Plant growth regulator PGR such as auxin, gilberellin, cytokinin, abscisic acid and ethylene are commonly used for the modern agriculture, horticulture and viticulture, including in okra plant. PGR are producing signal molecule within plant that exist in incredibly low concentration. From previous study, it showed that PGR responsible to regulate all aspects of the growth and development of plant, from embryogenesis, the regulation of organ size, resistance to stress, and the productivity development. However, although the several studies have been performed on this, the plant hormone that stimulate the growth of okra is not yet truly studied. Therefore, the objective of the study is to observe the growth of okra in the corresponding to the hormone growth regulator influenced by the chemical fertilizer. In the study, the okra plant were grown in the pot for 6 weeks and have given chemical fertilizer started at the week 4 and afterward for the growth. From the study, the plant EGH produced the highest number of leaves at the week 6 with the total number of leaves which were 9 leaves, and for the highest plant height was plant F with 36 centimeters. This finding indicate that plant hormone affected the growth of each okra plant with the help of chemical fertilizer. Hence, the chemical fertilizer could be considered as an alternative source to trigger the effectiveness of hormone on each plant sample. Characterization of each hormone is recommended for the future study in development of okra plant.
Assalamualaikum, my name is NURUL NADIA binti NAWI, S51668, and I will be presenting about the introductions. Okra plant or Abelmochus esculentus is an annual vegetable pot that develops rapidly up to 1.82 meter for the height, 10 to 20 centimeter for the long and wide leaf, 4 to 8 centimeter for the flower with 5 white to yellow petals and red or purple spot on the base and have several branches. Lady's finger is the common name of okra plant, and the plant belong to Malvaceae family. When applied, plant growth regulator PGRS and chemical promoting growth at early growth stage can alter the phenotype of okra plant. Plant growth regulators PGRS, also known as plant hormone, can be characterized as naturally occurring or synthetic compound that influence higher plant developmental or metabolic processes, often at low dosage. The 5 groups of plant hormones are consisting of auxin, gibberellin, cytokinin, abscisic acid, and ethylene. Auxin are the substances that promote growth in shoot elongations. However, they can inhibit the growth of lateral parts if the concentrations are high. Auxin also can be herbicide. Gibberellin or GA facilitates cell elongation, shoot production, and are involved in dormancy regulations. Cytokinin stimulate the division of cells by involving in the initiations of branching and stimulating buds. Abscisic acid regulate the dormancy of buds and seeds, prevent the growth of shoots, and if involved in controlling plant water loss. Ethylene are ripening agent which stimulate leaf and fruit abscission. In this study, the okra plants are grown in open surface area to figure how plants' hormone promote the growth and its development. The growth and development are quite fast, as it takes 2 weeks for the okra to start the germination of the seeds. The okra plants grew quicker with adequate amount of water, sunlight, and fertilizer. The effects of plant hormone have been well established in this study, as the plants, like leaf and stem, grew healthy but without the productions of the fruits. The okra plant's growth and development have not yet been completely studied. The plant hormones are responsible for promoting the growth and development of okra plants by releasing its hormone within the growth weed. This study found that plant hormones help okra to grow healthy along with the exposure to sunlight, enough amount of water, and the influence of the chemical fertilizers. The objective of this study is to observe the growth and development of the okra plants. The research project included the number of leaves and the heads of each plant obtained when grown together at the same time.
For the materials and method, the study was conducted at the Sri Manjung Perak Daud Rizwan. The analysis was conducted during the early sunny season to assess the performance of the okra from 22 November to 27 December 2020. For the preparation of the pot, okra seeds germinate best when soil temperatures range from 75 to 90 degrees Fahrenheit. And then we pick a pot for an okra plant with a diameter of at least 10 inches. We put in the pot in a position that receives at least 6 hours of direct sunlight every day. And then for the next analysis of our soil and PM, soil samples were taken at a depth of 0 to 15 cm before and after the analysis, and each pot was analyzed. Pottery manure samples were also obtained and analyzed in accordance with the standard protocol AOAC in 1990. Thin the okra seedlings to one plant for each pot, selecting the most vigorous of the seedlings after 2 or more sets of leaf mature. After that, water the soil as needed to keep the soil moist before the seedlings are established. After planting is well founded, dry-tolerant plants should only require weekly additional watering. We cut okra pots from the plant when they are 2.5 to 3.1 inches long, and then we check the plants with several days. Cultivate the okra, which is growth very quickly in hot weather. Regular watering is required during the germination and flowering periods. So deep soaking once every 10 days should be sufficient during long dry periods. When it comes to harvest and storage, we check the plants every day they start producing. Okra is a cut and come vegetable. So the pot first appear at the base of the plants so that by the end of the season we could be on our tiptoes. Experimental design. The experiment was conducted in a randomized full plot design, which is RCBT. And then for the collection of the data on plant head and leaf area were taken from 8 plants per plot at 1 week after planting. For the plant parameters it is including plant head and stem. The number of leaves were recorded in a table 1 and table 2 in the result. And the last one, we use data analysis. All data collected were subjected to analysis of variance ANOVA using statistics for agriculture scientists (SAS) package and a tool that is complementary. For example, for recording is used by Microsoft Excel to compile and record the data. And secondly, we use a t-test, which is a type of inferential statistic used to determine if there is a significant difference between the two groups. Means that may be associated with certain characteristic, which is when we don't put fertilizers and we put fertilizers, that kind of different in characteristics for the okra. And then finally we use the correlation approach to evaluate the relationship between the quantitative and continuous variables. That's all for the materials and method.
By the result obtained from week 1 to week 6, which is from 22nd November to 27th of November 2020, we can conclude that the highest amount of leaves on Eibosmochius esculentus plant are on Eibosmochius esculentus plant E, G, and H. Abomotius esculentus plant E, G, and H recorded 9 leaves on the end of week 6. Abomotius esculentus plant E, G, and H were under the influence of chemical fertilizer from week 4 to week 6, while the lowest amount of leaves on plant are on Abomotius esculentus plant A and D. Abomotius esculentus plant A and D only recorded 5 amount of leaves at the end of week 6. Abomotius esculentus plant A and D were not under the influence of chemical fertilizers from week 1 until week 6. As for the height of Abomotius esculentus plant, the highest height recorded in week 6 are on plant F. Abomotius esculentus plant F recorded 36 cm in the end of week 6. Abomotius esculentus plant F were under the influence of chemical fertilizer from week 4 to week 6. Meanwhile, the shortest height for Abomotius esculentus plant on week 6 are on Abomotius esculentus plant A, B, and D. Abomotius esculentus plant A, B, and D only recorded 30 cm of height on week 6, and Abomotius esculentus A, B, and D were not under the influence of chemical fertilizer from week 1 until week 6. From the result obtained, we can conclude that the application of chemical fertilizer on the growth of Abomotius esculentus plant can really help with the growth. As you can see, the growth on plants that are influenced by the chemical fertilizer are higher than the growth of plant without the application of chemical fertilizer.
Hi everyone, now I will share the discussion for the project in my group with the title Growth of Okra Influenced by Chemical Fertilizer. Here is the table that shows the number of leaf and plant height of okra plant within 6 weeks. Application of chemical fertilizer will influence the growth of okra plant. After week 3, plant E, F, G, and H are planted with chemical fertilizer for further study. All of the okra plants have the same number of leaves in week 2, but the plant height has a different data. Some of them are recorded as 8 cm, 6, and 7. Plant H, which has the lowest height, may due to the soil condition, where the plant watering and the amount of sunlight are constant. In week 5 and week 6, the number of leaves for plant A, B, C, and D are resulted in one increasing number, while plant E, F, G, and H are resulted in 2 increasing numbers. Furthermore, in week 6, plant F that applied with fertilizer has the highest height, whereas plant C that planted without fertilizer has the lowest height. Thus, this result proves that the growth of okra plants are influenced by chemical fertilizer. Chemical fertilizer can promote plant growth of okra plant by stimulating the regulators such as auxin, gibberellin, cytokinin, acetic acid, and ethylene. Both auxin and gibberellin play important role in plant growth because they are the hormone growth regulator that stimulate cell activation and elongation, and they also stimulate the leaf and stem growth. And here is the graph of number of leaves. In week 4, 5, and 6, we can clearly see that the plant without fertilizer has the less number of leaves, while the plant with fertilizer has a higher number of leaves. And also for the graph of plant height, the plant that apply with fertilizer has a good result compared to those without fertilizer. The use of chemical fertilizer has both positive and negative effect on plant growth and the soil. Chemical fertilizer are relatively inexpensive, contain a balanced distribution of nitrogen, phosphorus, and potassium, which are the three main essential nutrients required for optimal plant growth, and they are rapidly taken up by plants. However, the use of excess chemical fertilizer may lead to a number of problems such as nutrient loss, soil acidification and basification, chemical burn, and mineral depletion. Chemical burn is the situation where the plant turns into yellow and brown color due to the higher content of nitrogen in chemical fertilizer. And besides, overuse of chemical fertilizer will lead to the soil acidification. The high pH level of acid in soil will decrease the organic matter and hence affect the plant growth. Moreover, the use of chemical fertilizer also will cause soil depletion, where the nutrient and mineral in soil decrease. In order for plant to gain a healthy growth, people should avoid using chemical fertilizer in planting and gardening, but use organic fertilizer as a nutrient supply. Organic fertilizer such as animal manure or plant manure has a sufficient content nutrient, although chemical fertilizer contain more nutrient. In addition, organic fertilizer is more suitable for plant if compared to chemical fertilizer because it is safe and no harm done for environment and also the living organism. And the most important thing is the organic fertilizer can enhance the plant growth by improving soil texture.
Good day everyone, my name is THIVAGESH. My matrix number is S56156. So today I'm going to present about mini project report, which is my part is conclusion. Okay. Chemical fertilizers stimulate the hormone growth regulators and the growth of okra plant, which called as Abelmoschus esculentus. So plant growth regulators, which called as PGRS, are essential for Abelmoschus esculentus growth, elongation, specialization, and seed development according to the time. So Abelmoschus esculentus grown evenly for first 3 weeks, and the number of leaves are constant too. So then the number of leaves and height of Abelmoschus esculentus increased rapidly after chemical fertilizers are added. So these chemical fertilizers are act as a catalyst for Abelmoschus esculentus to grow faster and healthier. Okay, in my opinion, every plant need a healthy supplement like humans do, like for example fertilizers to enhance the outcome of a plant growth. So besides that, organic fertilizers are more effective for plant growth compared to the chemical fertilizers, as organic fertilizers are natural, which improve the texture of soil and increase of beneficial microorganisms. And thank you.